Thanks so much, Phil, for this nice introduction, and thanks everyone for attending this this great seminar. I've been very fascinated by the previous talk. It's always very interesting to see the many shapes that the topic of resistance can take depending on the system you're looking at. And I will, in the context of this talk, I will talk about viral diseases. I will more specifically talk about HIV, and again take up the thread of resistance, but look at it from a slightly different angle. Because what's special about an infection, of course, is that it can be transmitted, and with it, resistance can be transmitted, and that affects how we are discussing, how we're thinking about the problem. More specifically, I will talk about the use of personalized medicine or bioinformatic approaches in HIV research in the three following topics. I will first talk about the personalized approach to antiretroviral therapy and the immense benefits that this approach has had. Then I will talk about applying some of those approaches also to public health to understand the spread and the transmission of the virus. And finally, if time remains, I will talk about the impact of viral genetic variability on other outcomes, other than resistance. Most of the research I will be discussing is in the context of observational cohorts. In most cases, it's the Swiss HIV cohort study. In some other cases, it's cohort collaborations like the IDEA cohort collaboration. What is great about observational cohorts is already a point that has been made excellently by the previous speaker, is that in order to do research in this context, you need to combine genomic data, but also with relevant clinical and other non-genomic data. And that's excellently achieved in the Swiss HIV cohort study. It contains over 20,000 patients which have been followed longitudinally, some of them since the late 80s. And in those patients, over 20,000 viral genotypes are combined with all sorts of other data that may be relevant, and I will discuss some of that data in the next slides. In addition, a biobank allows always to retrospectively determine phenotypes of interest. So let me start with this basis on the first topic, namely on the personalization of antiretroviral therapy. I think this is really sort of an interesting case of personalized medicine occurring before the term was really widely established. If we look at the main developments, first just measuring viral RNA, then genotypic resistance testing, either in treatment experienced or treatment naive patients, you see that all of those developments have been taking place before personalized medicine was a big topic. And you will see in the next few slides the tremendous benefit that those approaches have brought. Since I assume that most of the audience does not have an infectious disease or an HIV background, let me just discuss some basics. Actually, from a bioinformatics point of view, it seems, at least on first glance, a relatively simple topic to a challenge to look at resistance in HIV these days. What you have the entire virus has a length of about 10,000 nucleotides. The gene which is responsible for resistance is the pol gene, and the relevant parts of the pol gene span around between 1,500 and 2,500 nucleotides. It's a relatively short genomic region where most of the variants responsible for resistance are located. This simplicity, however, can be also deceptive, because what makes the entire issue more complicated in the case of HIV is the large diversity of the virus within a patient it infects, and I will come to that later. So these nucleotide sequences can then be translated into mutations at given positions, which are known to affect different drugs. In order to understand the clinical impact of resistance, three basic concepts are important to keep in mind. The first is the genetic barrier of a treatment to resistance. The second is the fitness cost of resistance to the virus. And the third is, as I've mentioned before, the high viral diversity within an infected patient. So let me go through these issues with some rather old paper, but which make the point very well. So first, the genetic barrier. This is a concept which can be defined in different ways, either as the number of mutations the virus has to acquire to achieve high-level resistance, or as the number of substances in a combination therapy to which the virus is still susceptible, or from a theoretical perspective, most, I would say satisfactorily, as the probability that the virus achieves resistance during a course of treatment. And this latter approach has been, for example, done in the context of these conjunctive Bayesian networks, which some of you, I think in particular, Ashiel, if you work with Nico, you will know, have also an application in the context of cancer research. Independently of how you quantify the genetic barrier of a treatment course to resistance in a patient, what is important to keep in mind is that this concept is key to understand how the clinical situation has been improving over the last 20 years in HIV. The first aspect is the availability of new drugs and drug classes which have a higher genetic barrier to resistance. The second aspect is that the availability of new drug classes have allowed to tailor antiretroviral therapy in individual patients to maximize the genetic barrier to resistance. And the third is that in this context, the personalized medicine approach by sequencing the virus has allowed to tailoring that treatment very precisely. So that's the first concept. The second concept is particularly important in the context of the transmission of resistance at a population level. And here it's important to keep in mind that all these resistance mutations carry a substantial cost to the virus with them, which means that in the absence of treatment, those resistance mutations will tend to revert. And here you see the time scale of reversion, of the inverse time scale. And what you see is that most resistance mutations revert relatively slowly. They revert on a time scale of about 10 years, but some few resistance mutations revert much faster. They revert within one or within a year or less. And the background between the large variability of those reversion rates is that different resistance mutations have different fitness costs with them, as we could show a couple of years ago. The third concept which is important to understand antiretroviral resistance is that viral diversity, even within an individual patient, is one of the hallmarks of an HIV infection. And this is what you see in this graph. Here you see the average pairwise diversity within a patient as a function of the infection time in an infected patient. And you see that, you know, at least in chronic infections, you have diversities of 2% or more within a single patient. So if you pick two viruses from one patient, they will differ in they can easily differ in a couple of percent of their nucleotides. This also means that we may, if we just look at consensus sequences, we may not get all the resistance mutations which are relevant for a treatment, but we have to look at what are called minority variants. So variants which are at levels below 20%, typically with the currently used assays, variants at frequencies between 1 to above 1% can be detected. And what has been very nicely shown in this work led by Karin Metzner, who is also in our clinic, that these minority variants do indeed affect the odds for failing on treatment. So having such a minority variant increases the odds of failing on treatment by more than twofold. Nevertheless, it's an interesting discussion, which I will not go into here in detail, why those minority variants are not quite used in routine clinical practice. Nevertheless, taken together, this entire personalization of HIV treatments, or the achievement of treatments with high genetic barrier and achieving to tailoring those treatments to the virus infecting an individual patient, has allowed us in settings like Switzerland, so in resource-rich settings, to pretty much control the problem of resistance in HIV. And this is very nicely shown in two studies by which Alexandra Scherer did at our clinic. And here you see the number of newly acquired resistant viruses acquired over the years. Okay, and what you see is that this number has basically dropped to zero, not quite to zero, but almost to zero. And the few cases that remain occur in patients who have initiated treatment in the early phase of the epidemic. What you also see is that for newer drug classes, such as integrase inhibitors, there's virtually no transmitted resistance at all, which is observed in our settings. And with the concepts I have introduced at the beginning of this presentation, one can also get an understanding of what the reasons for this success are, but also what the limitations are. One of the reasons for the success, which is nicely illustrated in this graph, showing the level of transmitted resistance, so the frequency of resistance in newly diagnosed and untreated patients. Okay, this is shown here. And what you see very nicely here is that the introduction of new drug classes plays a potentially important role for this dynamics. So every time potent new drug classes have been introduced, we observe also a drop of this transmitted resistance at the population level. Note, though, that in contrast to the acquired resistance I have shown in the previous graph, the transmitted resistance never goes down, never disappears completely. So even though we don't have new cases of resistance mutations emerging in treated patients, at the population level, the pre-existing resistance mutations are still getting transmitted. Okay, but anyway, so that's the first component of control in resistance is new drug classes with a high genetic barrier. The second component is close surveillance and personalization of treatment in the patients which are treated. And this is very nicely illustrated by looking at these 4 drug classes. These 3 are the older drug classes, NNRTIs, protease inhibitors, boosted protease inhibitors, and unboosted protease inhibitors. And the shaded area, the gray shaded area, gives you the number of, or the sum of the virus loads, of log virus loads, in the patients who have been treated with those drugs, but where the virus is still allowed to replicate. And you see that in those patients, that for those drugs, even though treatment was very successful from a clinical perspective, it could, at the population level, never completely suppress the replication of the virus. So there was still some viral replication in the presence of those drugs. And that's always the source of new resistance mutations. If we compare that to the newer drug classes like integrase inhibitors, you see that treatment has become so successful and the tailoring of treatment has become so successful that the virus is virtually not allowed to replicate at all anymore within the patient it infects. Okay, and so if there is no replication, there can be no evolution, and that explains basically this fact that we have no resistance to these newer drug classes at the population level. However, one word or one reason for caution is this relatively slow reversion dynamics, which I have shown you before, because that means that once drug resistance mutations are there, they are very difficult to get rid of. And with those factors at hand, it's also easy to see that as soon as we are going to leave this setting that we have in Switzerland with a personalized approach, we will expect to see a higher problem of resistance. And this is exactly what happens as we go from resource-rich to resource-limited settings. Here you see a comparison we have done of drug resistance in 3 cohorts. One is the Swiss HIV cohort study. Another is the cohort at the IDI in Uganda. And the third one is a cohort in Lesotho. And you see that, you know, whether you look at this from the perspective of individual mutations or resistance levels to individual drug classes, you see that you see considerably higher levels of resistance throughout drug classes in resource-limited settings compared to what we see in resource-rich settings. And one of the main reasons for this is illustrated in a systematic review. Anthony Hauser, who is a postdoc in our group, has done this as part of his PhD, has done, and this shows you the level of resistance in patients failing a given treatment in Southern Africa as a function on the time these patients have spent on that regimen, which is a proxy also for how long those patients have probably been failing that treatment. And what you see is the longer time a patient has been failing the treatment, the higher the resistance level to that drug. So this is one of the main reasons why we see that difference here. So in the green and the red settings, patients have been failing treatment for a much longer time. The virus has been exposed for longer time to the drug and allowed to replicate in that situation. And therefore, the virus has been able to achieve higher levels of resistance. And now this is where the next slide is where is a consequence of this at the population level, namely resistance can be transmitted. So what we see throughout these resource-limited settings is that the prevalence of transmitted resistance mutation, so resistance mutation before you even start treatment, has been increasing exponentially and has been exceeding the WHO-defined threshold of 10%. We did actually some mathematical modeling work to capture this this development, and what we could show is that given the way treatment is administered in what is called the public health approach, so every patient receiving the same first line, second line, or third line regimen with relatively low switching rate, the same drugs being used, no resistance testing, and so forth. We could show that in that setting, this development of resistance is almost inevitable. So even if you would change one or two parameters of the model, you would still observe evolution or an increase of resistance at the population level. The big change has been fortunately achieved in those settings by introducing new drug classes. So as of 2017-18, the integrase inhibitor dolutegravir, which I have mentioned before, has become available at low cost in those settings and is being rolled out through Southern Africa and also other settings. And as we expect, as we have modeled here in this context, we expect that this introduction will at least resolve the issue of resistance to the older drug classes. So it will allow to control the resistance levels of the older drug classes. What is, however, very much an open question is how robust this new treatment classes will be on the context or in the setting of a public health approach to treatment with a relatively sparse surveillance, allowing HIV in millions of patients potentially to replicate in the presence of dolutegravir. So this is one of the big questions for the future: how evolution-proof, if you will, those therapies are under the more challenging condition of in resource-limited settings. Let me nevertheless return to a more positive message, namely just illustrating on one illustrative patient from our cohort the tremendous success of antiretroviral therapy. So you see that these, you have a patient who has been treated, there's a patient who has been treated early as the combination therapy regimes have become available. The viral load has been suppressed for decades. So for decades, there has been no noticeable replication of the virus, and the immune system, as shown here in the CD4s, has recovered. The big next achievement for these patients is to obviously cure them of the infection, because as you see from those occasional blips of viral load, even though viral replication is successfully controlled, you cannot stop treatment, because whenever you would stop treatment, the viral load would go back to its initial level. And this is one of our research interests, is to look not only at replicating virus, but to look at integrated virals, proviral DNA, the viral integrated in the level of integrated viruses in the host cells. And what you see is that this level of the integrated level of virus, it decreases. We did that in over 1,000 patients of our cohort, and we measured the level of integrated virus at 1.5 years, 3.5 years, 5 years, and beyond, and looked how fast it declines. And what you see is both a good and a bad thing. you see the level of integrated virus, so the level of virus which is integrated into the human genome, it decreases significantly over time, but you also see that it decreases over time, but you also see it decreases with a diminishing slope, okay? And if you look at the slopes, you see after the distribution of the slopes, you see that it's, it may be on average negative, but not at all for all patients. And this is again one of the hallmarks of all infections. Whatever type of phenotype you look at, you always see a tremendous inter-individual variability. And we can now look at the clinical determinants of this variability. But if time remains, I'm afraid not, we could also make the case that the viral genotype plays an important role of whether one patient has a decreasing level of integrated provirus or an increasing level of integrated provirus on the successful antiretroviral therapy. Let me now switch gears for the last minute and briefly illustrate how these genetic resistance tests or these viral sequences can not only be used to understand resistance and therapy success, but how they can also be used to understand the transmission of the virus at the population level. The principle of this is actually quite simple. It's what you do in molecular epidemiology of infectious diseases. So as a virus is transmitted in the population, it keeps evolving. Therefore, if we do a phylogeny of the sampled viruses from the population, then the structure of that phylogeny will reflect the structure of the transmission chain. We have used that in many ways to characterize the HIV epidemic in Switzerland and also in other settings. Essentially, this approach allows you to characterize the structure of the transmission chains, to infer who infects whom, and which groups, which subpopulations are interacting epidemiologically. I want to illustrate this approach in one recent study where we have tried to understand what determines that some of the sub-epidemics that we occur seem to be growing while other sub-epidemics do not grow. So if you think about the entire HIV epidemic in Switzerland, it consists of different micro or sub-epidemics, if you wish. Some of them are relatively inactive, so they are no concern for public health, while others will grow rapidly, and those are the ones we would need to target if we want to eliminate HIV transmission one day. The idea of doing this is the following. So we take all sequences in the cohort, we construct a phylogenetic tree with those sequences. We identify transmission clusters in those sequences for a given year, and then we repeat the same approach the next year, and we look for one of those phylogenetic clusters. How many new sequences or how many new patients has that cluster acquired? And then we try to correlate this outcome. So the outcome is the growth of a phylogenetic cluster, and we try to correlate that with how many infectious patients this cluster has had and what the risk behavior of those patients has been. And if we do this approach, we see that over the years, we distinguish here patient clusters that grow, which are the open squares, from clusters that do not grow. And we see clusters that grow have both higher levels of infectious patients, okay, and higher risk behavior. So this essentially shows that treating patients and making them non-infectious indeed has also a beneficial effect at the population level of stopping viral transmission. However, what we also see at this in this analysis, that a large number of transmission clusters that do grow, these are the greens one here, have no single infectious individual in them. And that's exactly the cluster that we would want to target, because those clusters indicate that there are in the same transmission network infected individuals who have not been diagnosed or treated and therefore can still continue to spread the virus. I see I'm out of time, so I will have to skip the last topic, the impact of viral genetic variability on phenotypes other than resistance. Suffices to say that the viral genotype matters actually for a surprisingly large number of phenotypes, ranging from the immune response, the breadth of the immune response you see in infected patients, to whether a patient has, for example, neurocognitive symptoms or not. But I'm happy to discuss those topics later on. With this, I would like to finish. I would want to acknowledge all the people who have been involved with this. This is a hugely collaborative research involving clinicians, bioinformaticians, epidemiologists, lab people, and so forth. I would like to thank everyone, and I would like to thank you for your attention.